And privilege as president to be able to introduce the George Pickering Lecture. And this year I'm very pleased to be able to introduce my good friend and past president, Professor Maurice Brown. And Maurice is, I think you'll agree, one of the intellects in the world of hypertension. He's done enormous things, and through the last, probably 20 years, his work has formed a major part of the British Hypertension Society scientific meeting. Maurice is an incredibly productive researcher. He has done so many things, we just don't know how he manages it. He does randomized trials, wet lab work, genetics, and he also runs an amazing team at Cambridge. If you've met his team, they're all incredibly smart, witty, blonde, and and he manages to get them to produce amazing work, and they're all very highly supportive of him as well as he is of them. In fact, his nurses wanted me to point out that they want to be known as his foxy girls, so there you are, I've said it. Maurice is the epitome of what we'd all like to be. I don't know how he manages to do it. His CV is a great testament to his productivity. The executive, when we were deciding on the Pickering lecture this year, had no hesitation. We just stopped when we thought of Maurice, and that was it. It was unanimous, and I'm very, very pleased to be able to ask him to give this year's Pickering lecture. Maurice.
Well, Tom, I'm speechless. Can I stop there? That is my second slide. I'm just going to go back. There we go. It is a wonderful honour to be standing here, and I'm enormously grateful to the Society and in particular to the President. Among many reasons why I've felt a tingle in my bones of the anticipation of this moment. Perhaps the strongest is that I not only come from Edinburgh, but I used to come to the Christmas lectures in Surgeons Hall when I was a child, brought here by my mother and taken home by my father, who had premises across the road that are now the site of the Festival Theatre. I hope they may both be looking down, and that I will not disappoint them and the many others who have helped me to come here. As the title of my talk suggests, I'm going to dip in and out of both clinic and laboratory. And indeed, my good fortune has been to witness the virtuous circle of science and medicine, hopefully to the benefit of patients present and future. So please enjoy this story. I think I've only missed one meeting over the 34 that we've had, and I always smile at these simple questions remain unanswered or controversial. Are they even the same question? It's at the least piquant that a Sassenach comes to Scotland to rerun a battle between two English lords. Baron Platt, who brought the London Royal College of Physicians to its current abode in Regent's Park, is regarded by history as the loser, but was he partly right? Platt's view was that hypertension is a discrete entity different from the factors regulating normal blood pressure. Sir George Pickering, whose memory we honour today, was professor at St Mary's and then an old university on the road from London to Stratford. He dismissed the notion that hypertension was a single entity, and tongue-in-cheek can be said to have predicted the gooey world of genome-wide association studies. So there's a summary of the Pickering view. As nicely redrawn here by Sandosh and colleagues, the Platt-Pickering dispute can be represented by frequency plots. First, the Platt view of hypertension, and then the Pickering. Sir George was, of course, right that there are multiple causes of hypertension and that blood pressure is a continuous distribution. But was Platt right if the inference is that single causes are common and sometimes curable? Is it a coincidence that the phenotype on which I will be measuring today, namely hypokalemia, trips off the autoanalyzer? Or is it the tip of the iceberg of many yet to be discovered single causes? Well, blood pressure is much easier to define than hypertension, although paradoxically, the answer has had even less influence on practice than Pickering's, or I should say much less influence on practice than Pickering's answer to what is hypertension. Every GCSE or higher student knows that pressure just is force over area. And John Lara, Poiseuille, and Guyton have derived the useful vasoconstriction volume diathesis from the equation shown here. Of the several heroes in my story, John is just young enough, in his late 80s, for me to have interacted with him. And one of the proud moments during the last year was to be invited to take part in his Festschrift and to receive a touching note of thanks, which hopefully you can't read, from his wife, Jean Seeley. Well, not only every GCSE student, but also every gardener among you knows what pressure is. There are just two ways to increase the jet of water from this hose. You can either kink the end, reducing area, which is what the sprinkler does, or you can turn up the tap, increasing force. In the circulation, force is essentially the amount of salt and water. Loss of salt leads to a compensatory reduction in area of resistance vessels in which renin is the main player. Well, in Edinburgh, as the home of the great empiricist David Hume, I can acknowledge that all the rationalist arguments I've just shown you came, in my case, entirely post hoc. When I designed the rotation study that led to ABCD, I was an avid disciple of the genetics revolution, heralding, if not one Plattian gene, at the most 5 or 6. And with the brashness of youth, I thought that if I rotated patients through the 4 main classes available to us, I would find 4 sets of responders with the genes not far behind. The 2 pieces of serendipity were, firstly, that in selecting low-risk patients in whom I could design a study of a month-on, month-off, I chose a young age group. And the second bit of serendipity was that the 4 main drug classes start with the 4 letters of the alphabet. Well, the first surprise was that in this young set of patients, all aged less than 50, the average response to all drugs was not similar or equally good. And it was plain that the drugs acting on the renin system, A and B, were almost twice as effective as those which work, at least in part, by eliminating salt. The next set of data, even 15 years on, is still a challenge to present. Suffice it to say that the messages are the widespread variation in response influenced by these frequency plots, which we take for granted today, but were almost heresy in the late 1990s. And the second was that for all the possible correlations between pairs of drug response, with the y and x-axis on this graph being fall in blood pressure, only 2 correlations were significant. The one between the 2 drug classes that work by blocking the renin system, and the one between the 2 drug classes that work in part by eliminating salt. So these points were highlighted in the abstract of the paper, in which I concluded that most patients would not achieve their target or their best blood pressure response unless they rotated through all the options. And secondly, that one might be able to shortcut such a rotation by a rule which said, firstly, although we have 4 main classes, there are actually only 2 categories of drugs. And secondly, that the choice between these could largely be predicted by a patient's age, working as a surrogate for plasma renin. And I was influenced by a work from Stan Peart and Tom Mead showing a slow decline in renin with age. So this is the first version of the ABCD, which I slipped into the discussion section of the Lancet revision. And it's the parent, if you like, of the current AC rule. Exceptions to rules are often informative, and the original rule had a switch stage in which patients unresponsive to the drug class typical for age were switched to the alternative. And anticipating what follows in this talk, I now believe that the young patients who do not respond to A or B are the ones in whom we could most usefully measure the plasma renin. Now, at the stage that in the 1990s, when ABCD was first put forward, it was in truth more of a prediction than a rule, since we had little evidence yet that the older patients would respond better to CD than AB. But my good fortune was that many large trials were in progress which would answer the prediction. And I'll just show you a few of the main ones. Firstly, value, which was a comparison of an A drug versus a C drug. And in the first few months of the trial, the fall in blood pressure when patients were still on monotherapy was about 6 millimeters more on the C drug in these older patients than the A drug. Then ASCOT followed, and although the y-axis is more squashed than for the value study, once again we see that the C drug in blue is more effective than in this case the B drug in red. And then finally, the largest of all outcome trials, ALLHAT, and we see that the A drug, lisinopril, in green, is less effective than the red or blue, namely the C or D drugs. Now, given the weight of evidence in favor of the two patterns of response, and more importantly, the laws of physics which underlie the duality, I've naughtily likened the deniers to the Flat Earth Society, but even they, in January of this year, saw the light over the curved horizon, and you see that their guideline acknowledges the duality, whether on ethnicity or age. I have to say that although I'm all for a redefinition of youth, I'm not quite sure what the evidence base is of changing the threshold to 60. And so now to renin, the first hormone to be discovered in 1899, but yet to enter mainstream medicine. 10 years ago, we measured plasma renin in some 850 patients coming to their primary care, their general practice. And in some ways, the main finding of practical interest is the width of the x-axis. This means that in looking at the extremes, whether the patient is a high or low renin patient is not going to be influenced by the stress, for instance, of trying to find a parking place at the hospital. In the extreme in which I'm most interested, low renin patients reflecting salt and water overload, the only main confounder is the use of beta blockade, which works by suppressing the plasma renin. Well, renin, we think, explains the variance of response to therapy, but in truth, its measure as a predictor is still largely unproven. And of most interest are the patients with resistant hypertension, in whose place in the volume vasoconstriction spectrum can no longer be guessed or predicted from their age. So this you will recognize as a redrawing of the current NICE AC algorithm, and I'm now superadding the measurement of plasma renin with the hypothesis that we can predict those who will respond better to beta blockade at one end and a change or additional diuretic at the bottom. And alpha blockade is slotted in the middle. Well, this is a hypothesis at the moment, and stay where I want to. And one of the successes of the British Hypertension Society is that we have recruited 350 patients into a trial, Pathway 2 trial, and in a year's time, we will have the answer to the questions, does plasma renin predict the best response? And is one of the drugs, probably spironolactone, more effective than the others in treating resistant hypertension? So to return to the algorithm, and as the title of my talk suggests, can renin do better than just predict best treatment? Can it predict patients whose hypertension we can cure altogether? At the high end of the renin spectrum, we might consider, for instance, sending a patient to renal angiography, looking for renal artery stenosis. But the rest of my talk is going to be concerned with finding the aldosterone secreting adenomas, which are most likely to turn up in patients with a low renin, despite being on multiple drugs, A plus C plus D, all of which ought to have been elevating plasma renin. Well, this is the 60th anniversary, as you heard from some of my students yesterday, of Conn's discovery of his eponymous tumours. And his index patient was a young woman with hypertension, low potassium, and alkalosis. The recently discovered hormone aldosterone was at a high level in the patient's urine, and Conn sent her for bilateral adrenalectomy. and the rest is history. Ironically, the large size, 4 centimeters, of the tumour was atypical and may have held back the search for the more typically sized, smaller tumours. The best estimate of the true prevalence of Conn's adenomas comes from this study of Gianpaolo Rossi in Padua, who looked at newly diagnosed hypertensives and reported that almost 5% of patients had a surgically curable adenoma. For reasons that will become apparent, I think even this may be an underestimate, since the smaller zona glomerulosa-like aldosterone-producing tumours are underrepresented in subsequent genetic analyses from the same group in Padua. Well, one of the major reasons why such a common condition is undersourced and underdiagnosed is the difficulty of the downstream tests, particularly the adrenal vein sampling. This slide illustrates a typical patient with an obvious adenoma on the MRI scan, subsequently removed. But few hospitals have the radiologist with a greater than 50% chance of cannulating both adrenal veins, particularly the small right adrenal vein entering directly into the IVC. And at this 50-50 rate, few patients ever enter the diagnostic pathway. So let me roll the clock on to the typical patient whom we're seeing now and diagnosing in Cambridge. And I suspect there's not much in the information I reveal at the start of the slide that would set many passes racing. But the low plasma renin at 7, despite use of high-dose angiotensin blockade, 32 milligrams of candesartan, is enough for us to send this patient for a CT scan of the adrenals. The axial view was not definitive, but the coronal reconstruction that I always recommend shows a small but definite adenoma. The game changer is the ability to look at small or ambiguous masses and say whether they are functional, and this patient proceeded to adrenalectomy and cure of his hypertension. Well, carbon-11 metomidate that was used in that scan is not a new tracer, and metomidate itself has been around since the 1960s, together with its first cousin etomidate, the ethyl rather than the methyl imidazole. They're both effective anesthetic agents, and both were incidentally found to inhibit cortisol production and therefore to be high-affinity nanomolar ligands for the enzymes that make both cortisol, CYP11B1, and aldosterone. I'd like to say that our contribution here was to develop a PET-CT which compared the success of the non-invasive imaging with adrenal vein sampling. And the main reason for putting this slide up is to make sure that I pay tribute to the many and multidisciplinary people who took part in the study, and in particular to Ireland Mackenzie, who wrote much of the application of the British Heart Foundation that funded the study. So what I'm going to do next is, before I come back to the use of the PET-CT, is to show 2 patients who incidentally turned out during our pathway study to have a curable cause of hypertension. And as the imaging study, or after the imaging study finished, we had a gap before funding was available for routine PET-CT, so both these patients, in fact, had adrenal vein sampling. The first patient came into pathway in his late 40s, had obvious hypertension despite being treated with 4 drugs. There was nothing in his baseline electrolytes to make us suspicious, but on the renin and aldosterone, which is measured at the outset in all patients in pathway 2, he has a low plasma renin, high plasma aldosterone, which makes the diagnosis of primary aldosteronism. At the end of pathway 2, this slide shows the blood pressures, systolic in orange, diastolic in blue. We're allowed to unblind each patient's best cycle, and it's evident for this patient it was cycle 3, and his most effective drug was spironolactone. So the patient proceeded to have a CT scan of the adrenals, which I've outlined in red. The scan was reported as essentially normal, but as you will have gathered by now, I don't like taking no for an answer, and for me the arrowed area was almost certainly an adenoma. So the patient went to adrenal vein sampling, and indeed his 100-fold ratio or gradient from right to left is one of the highest that we've seen between sides. So he proceeds to surgery, and the picture shows the small tumour, a high-tech device we have in Addenbrooke's for measuring the size of tumour, something around 1 centimetre. And not only is he almost cured of his hypertension altogether by the removal of his adrenal, but he's one of the 30% or so in whom surgery has transformed him by giving him a new lease of energy. So I would like, just in case you think he is a one-off, to show you one other patient from Pathway who has also been cured, and say there are several others awaiting surgery. This patient, too, was in his late 40s, presented with, on 4 drugs with no clue in his baseline electrolytes. We had a relatively low plasma renin and an elevated plasma aldosterone. In the change of drugs before entering Pathway, particularly the withdrawal of beta blockade, left him with a renin that was still suppressed. But I'd like to point out that the change to amlodipine would have masked the diagnosis if we depended on finding a high aldosterone. And this is one reason why I do not think aldosterone is essential to making the diagnosis of primary aldosteronism. So he too proceeds to a CT scan. Once again, the axial view not absolutely clear-cut, but he clearly has an adenoma on the coronal reconstruction. Again, he has adrenal vein sampling, which shows an obvious difference between right and left. And he proceeds to laparoscopic surgery, where a 6 millimeter adenoma, most of this section is actually fat, here's the adenoma, is removed. And once again, this patient is completely cured of his hypertension, despite being on 4 drugs before he met us, and he's also cured biochemically. And at this point, I need to pay tribute to some of our Pathway nurses, Jackie Salisbury and Schumann, who, after patients are finished with the study, undertake the extra work and I hope pleasure of seeing patients go from multiple treatments to cure. Well, I'm going to change gear now. I hope, stimulated by these 2 patients, you are curious to know how come such aldosterone-producing adenomas can be so small, so common, and yet cause such severe but curable hypertension. And I need to start with a painless reminder of adrenal histology. On the left is a summary of the different zones of the adrenal cortex and the hormone that each makes and what they do. On the right, I illustrate some of the differences in histology between the outer 2 zones, because this is what gave us the initial clue to the presence of 2 different types of tumour, and why paradoxically the classical Conn's tumour illustrates or resembles the cortisol-secreting cells rather than the cells which normally make aldosterone. Well, Lady Luck, on whose intervention I've remarked previously, visited us again in the form of Rick Lifton's landmark paper in 2011 describing somatic mutations in a potassium channel, KCNJ5, in some 30% of tumours. This is a fascinating enough discovery on its own, but John Funder's editorial, which I call curmudgeonly clairvoyant, gave us the idea that Rick's KCNJ5 mutation was positive in tumours at one end of a phenotypic spectrum. This, which I call my boxing ring diagram, shows that there is indeed a pronounced genotype-phenotype relationship that is apparent when we plot the size of a tumour against the age at which patients present. In the top left red corner, we find the classical younger women in circles, and the red colour means these are the ones with KCNJ5 mutations. When we look at the histology of their tumours, the cells resemble the lipid-rich cells of the zona fasciculata that you've just seen in my previous slide, and they have high levels of the hallmark ZF enzyme, ß-17A1. In the right-hand blue corner, the patients are older at presentation, often present with resistant hypertension, and are more often men than women. And the histology of their tumour shows the small cells more like the ZG cells that normally make aldosterone, and these tumours have a relatively low level of ß-17A1. Well, we joined up with Dick Gordon and Michael Starras in Australia to add some samples to our collection so we could show robust statistical significance of this phenotype-genotype relationship. And about the same time, Alina Azizan, who did much of the work I'm telling you about, also spotted an almost categorical difference in these ZG-type tumours with the presence of previously recognized spironolactone inclusion bodies only in the ZG-type tumours. Yet the notion that we have here a different subset of tumours has remained controversial, and that was one of the reasons why we wanted to proceed to finding whether there was a distinct mutation that marked these ZG-type tumours as different from the classical ZF. And in order to pull an extreme group out of this effectively continuous spectrum, we used percentage of compact cells as our phenotype. And on this slide, you see the percentage of compact or ZG-type cells plotted against whether the patients were KCNJ5 positive or negative. And you can see that none of the KCNJ5 positive tumours have a high proportion of compact cells, where there appears to be a distinct group of patients whom we now took 10 of and sent for exome sequencing. And for those of you not familiar with this technique, it's a way of sequencing every coding region of every gene in the genome, in which we compare the tumours and the normal germline DNA at a cost of about £1,200 to £1,500 per pair. Well, the results of this experiment were amazing. Here are the 10 samples that we sent, most of them male, all of them less than 2 centimetres in size, some less than 1 centimetre, and interestingly, in many cases, multiple tumours, although only one of them ever had the mutation. And we found that only one of 2 commonly recognised genes provided the mutation in 9 out of the 10 patients. One is the alpha subunit of sodium-potassium ATPase, and the other is a L-type calcium channel, first cousin of the CAV1-2 calcium channel that is the target of drugs like nifedipine, with 4 separate mutations being found in 5 patients. Well, the next thing we did was to ask whether these mutations we'd found were actually functional, and we turned first to Paul Nissen and Hanna Poulsen in Aarhus in Denmark, who'd spent their lives working on sodium-potassium ATPase. You can be as ignorant as I was at this stage about so-called current voltage plots, and still appreciate the gross effect of the mutation in sodium-potassium ATPase. The dominant direction for current through this pump is outwards, with the sodium being exported from the cells, providing there's potassium outside, which conventionally is shown as current above the x-axis. Whereas by contrast, when the mutant is transfected into the frog-oocytes, there is no current above the line at physiological voltages. And you may also note that the scale of the y-axis, the voltage, is some 10 times greater than, sorry, that's current. The scale of the current is 10 times greater for the mutant than the wild type, meaning that there is a massive inward leak of cations. For the calcium channel, we were helped both by Annette Dolphin at University College in London and Jörg Strisnik and colleagues in Innsbruck. And I'll start by showing the structure of the channel, which is 4 repeating domains of transmembrane segments, 6 segments in each domain, with the calcium pore always between segment 5 and segment 6, and the mutations are each shown as coloured balls. Actually, once we'd done replication, we rose to 8 of these mutations, again, all in or near the calcium pores. For simplicity, I'll just show you how 2 of the mutations work, because they varied in their effect, and once again, this is a current on the y-axis versus voltage on the x-axis plot. And compared with the wild-type or the normal channel in black, you can see that the mutations have shifted the curve to the left, meaning that the channel is more likely to open at a relatively small level of membrane depolarization. Subsequently, an MPhil student, Catherine Zeev, studied the mutation in human adrenal cortical cells, showed that it increased aldosterone secretion, and that this increase was reversed by an experimental blocker of the CAV1-3 channel. Well, at this stage, I obviously want to thank many people. I think this was the most exciting phase of my career, and I owed help in particular to Elena Azizan, who I've already mentioned, who was a PhD student and then postdoc, many others in my own lab and others in Cambridge, but also to a sort of mini Europe of collaborators who helped us both with the functional studies, as you've seen, and with replication in independent cohorts. This was really great fun and very, very exciting. In the same year as we published our results, we were not alone, and 2 other groups published in the same journal. The German group were actually ahead of us while we were waiting to find function for our calcium channel, whereas Rick Lifton's paper appeared in the same issue as ours. But I think we were the only ones to recognize the clinical phenotype associated with the new mutations, and in a way this has been the most exciting feature, the immediate clinical impact, allowing us to recognize more patients with small tumours and the new mutations. And that's what I wish to illustrate in a moment. But because this still remained controversial, we felt we needed to provide a little more evidence, and to do this by finding at least one or several genes which are exclusive to the smaller ZG type tumours and which are normally expressed in ZG. Now, some of you will have heard a fuller version of this story in Ada's talk yesterday that won her Young Investigator Award, but it's so nice, let me just show you a few of her slides. We performed a microarray comparing what we now call the ZG-like tumours with the new mutations against the classical ZF-like tumours. And the results are shown with the genes which are differentially expressed between the 2 types of tumours on the vertical axis and each of the samples on the horizontal axis. Red means increased in abundance and blue means reduced. The top hit was a gene called Nephronectin, or NPNT, which was more than 12-fold more abundant in the ZG-type tumours. This, as its name might suggest, is a gene previously recognized in the kidney, as illustrated here with the G standing for glomeruli, and the high power view, which comes from public domain immunohistochemistry, suggests that it's a matrix protein. So the question is, will we find it to be selectively expressed in the zona glomerulosa of human adrenal? And the answer is spectacularly yes, as shown in this rather beautiful immunohistochemistry performed by Ada. And the next question is, does this staining also selectively stain the ZG-type adenomas? And the answer again is clearly yes. This has been done in 4 pairs, illustrated by one of them here. And quantitative RNA analysis confirms the much greater expression of NPNT in the ZG-type tumours. So for this sort of reason, we are pretty satisfied now that there is a truly separate group of tumours. But the story gets better, because on closer inspection, we see a potential role for NPNT. It's not just a marker. It outlines the glomeruli from which the zona glomerulosa takes its name, but whose function has never really been studied. And those of you who heard Ada's talk yesterday will know that we now have some ideas about a role in calcium signaling between the cells in each glomerulus. Suffice it to say that Ada went on to show that NPNT drives aldosterone secretion, but I'm just going to observe that whereas in a normal adrenal, we have this selective expression of NPNT in zona glomerulosa, next to a Conn's tumour where there's negative feedback on aldosterone secretion, the NPNT virtually disappears, and this is confirmed quantitatively in the RNA measurements. Well, many of you will be relieved to know that I'm going to leave the laboratory for most of the rest of my talk and now show how these results have influenced our practice. And we took the results back to the clinic to ask the question, can we find these smaller APAs which our results are predicting, and use our PET-CT in the process. The first is a patient who I first encountered when he was in his 30s, and at that stage I was not yet aware that a low plasma renin at that age is virtually diagnostic of a Conn syndrome. Subsequently, the penny dropped almost literally when he went on a diuretic rotation study and dropped his potassium on the thiazide. However, the CT scan was ambiguous at best, and he did not lateralize on adrenal vein sampling. However, when we went back to him with PET-CT, this nodule clearly lights up, and he proceeded to surgery where he had a definite sub-centimeter adenoma, and this is positive for one of our calcium channel mutations. My next example is someone with clear-cut Conn syndrome, presenting at a young age with severe hypertension, hypokalemia, suppressed renin, high aldosterone. However, the CT again is ambiguous. It was reported as a sausage-shaped adrenal, and once again, he did not clearly lateralize on the adrenal vein sampling. However, the PET-CT lights up a nodule at the bottom of the adrenal, and when he comes to surgery, this nodule actually looks different from a string of nodules which the adrenal turned out to contain. And on biochemical analysis, only the nodule which was positive on the PET-CT scan is strongly positive for the enzyme which makes aldosterone, both at the RNA level. This is the only one that has the mutation in sodium potassium ATPase, and at a protein level, when stained for the enzyme that makes aldosterone, only the nodule that was positive on the PET-CT is positive for aldosterone synthase, whereas one of the other nodules, this one up here, is positive for B1, the cortisol enzyme, telling us two very useful points. One is that the PET-CT, surprisingly, is not very good at seeing cortisol. It's very useful at explaining a low background of a normal adrenal, but it also explains the negative adrenal vein sampling, because I think the high cortisol coming from the nodule here was cancelling out the high aldosterone from the nodule at the tip of the adrenal. Well, up to this point, we'd used the PET-CT in patients where there was some anatomical basis for expecting an abnormality. But perhaps most exciting is the discovery by PET-CT of adenomas in adrenals which looked apparently normal on the CT or MRI. And my first example is a 55-year-old man with severe hypertension on 4 drugs. There's not a lot of information about the first part of his story when he was seen privately, but the PACS record shows that he was correctly suspected of a Conn syndrome because of the suppressed renin and elevated aldosterone, but he had a normal adrenal on MRI. He successively had an MR angiogram of his renal artery and formal renal angiography, and for reasons which aren't quite clear, he was then proceeded to renal denervation, perhaps as a more evidence-based strategy than further investigations of his adrenals. But unfortunately, denervation, if anything, led to a rise in his blood pressure from about 170, 110 to 190, 120, and the patient disappeared in some disgust. Fortunately, and perhaps not wholly coincidentally, he also had paroxysmal atrial fibrillation, and following his successful ablation for this at Papworth, he was sent back by the cardiologist for investigations of his Conn's syndrome. Well, as I've said, up until then, I would probably have used adrenal vein sampling in the absence of an anatomical lesion, but this patient was in no mood for further invasive investigation, and so he had the PET-CT. And I think you'll agree the result is beautiful. Here you can see a hot adenoma outlined by the almost cold limbs of the normal left adrenal. And with that wonderful instrument, the retrospectoscope, we can go back to the original MRI scan. Oops, sorry about that. The original MRI scan, and on just one cut of one sequence, you can see a tiny little adenoma peeping out, probably also present on the baseline CT of the PET-CT. And just to enjoy them, here are the coronal views of the same patient. So he proceeded to adrenalectomy, which confirms the presence of a small adenoma, and within hours of this being removed, his blood pressure has fallen to normal. Well, the initial blood pressure response isn't always that meaningful, but 1 month later, he's almost normotensive and cured biochemically. And because I knew I was going to talk about him at this meeting, I asked him for a follow-up. He dutifully sent me some home blood pressure readings, but also his rather spontaneous, lovely comment on his symptomatic improvement. Well, this patient, I think, was in a way fortunate that we didn't do adrenal vein sampling, because in a number of other patients with these ever smaller adrenal adenomas, adrenal vein sampling proves negative, and especially if the patient is one with very little periadrenal fat, it can be very hard to see the adrenal, and the PET-CT isn't always quite as what I may call slam dunk as the patient we have just seen. So here's an example of a young woman, adrenal reported as normal, probably positive PET-CT, but normal adrenal vein sampling. With the retrospectoscope, again, I think you can probably see there is a small adenoma there. And then my probably youngest ever Conn's patient, a current student, again, a possible adenoma in left adrenal, negative adrenal vein sampling, but again, you can see an adenoma lighting up between slightly colder limbs of the normal left adrenal. Well, fortunately, we do have a third modality for these ambiguous patients. My colleague in Cambridge, Nick Carroll, has been doing endoscopic ultrasound with fine needle aspiration for almost 10 years for a range of suspicious masses, including larger ones in the adrenal. And I was unaware in my anatomical naivety that the left adrenal sits just a few millimeters from the stomach. And here you can see the left adrenal adenoma of the first patient. So here's the possible adenoma on the MRI, easily seen by endoscopic ultrasound with the normal left limb disappearing northwards up here. And a needle is put into this, and here is the fine needle aspirate staining positively for CYP11B2. Well, in the future, we plan to return to such patients for endoscopic ablation, and this may even come to replace surgery as the intervention for patients. And I was hearing last night that a comparison of percutaneous ablation and surgery is already underway in Hong Kong. Well, I think we've only touched the tip of what's possible with the PET-CT looking for Conn's adenomas, and the group who may benefit the most are the Afro-Caribbeans. And I'm sure most of you know that typically the Afro-Caribbeans, even at young age, have a low plasma renin, and they are a salt-dependent type of hypertensive. But less well known is that the plasma aldosterone, as in this very nice data that Phil Hovienchik and colleagues have let me have, the aldosterone is slightly and significantly higher, despite the lower renin, than in matched Caucasians. And rather than search for a genetic cause in the kidney, the sort of obvious question is, do these patients have a sort of epidemic of small adrenal adenomas? And Phil and Kennedy Cruikshank have started sending us a number of the Afro-Caribbeans, and the results are indeed amazing. Here's the very first patient that Kennedy sent me. No doubt that he has a hot adenoma in the left adrenal, and again, with the retrospectoscope, you might be able to see it on the CT. So I'm going to take you back briefly to the lab to look now at the question why we think these adenomas are so common and yet so small and easy to miss. And my theory, which is speculative, is that constitutive activation of aldosterone production rather than proliferation of the cells gives the ZG cells a selective advantage. And the reason which stimulates this is that most of us are eating too much salt, which tends to make the ZG cells disappear. So a number of people with good antibodies to the enzyme that makes aldosterone, particularly Celso Gomez-Sanchez, have shown quite how sparse the expression of aldosterone synthase is in the normal human adrenal, putatively because we're switching it off with salt. And 10 years ago, it was shown that if you delete the enzyme genetically in mice, the cells which make aldosterone from the ZG largely disappear. On the right-hand side, in the genetically deleted mouse, you can see that the ZG cells in yellow have migrated from the ZG to the ZR medullary boundary, and then in the lower right-hand panel, these same cells are staining positive for the marker of apoptosis on tunnel staining, whereas in the control normal mouse on the left, the ZG cells have no apoptosis. So do we find apoptosis in human adrenal? And the answer is yes. In this nice work done by Lala House-Sheikh and Nikki Figg and Martin Bennett's group, the ZG at low and high power of human adrenal is positive both on the tunnel stain that you just saw in the mouse adrenal and on an alternative measure of apoptosis, cleaved caspase, giving this brown staining. So my final bit of lab work and piece of evidence that human adrenal ZG is very unusual compared to probably most other species, probably sowing the seeds that allow mutations to be selective that switch on constitutive aldosterone production, is our finding of multiple genes which are uniquely expressed in the human zona glomerulosa and unusually seem to be telling an endocrine gland to switch its hormone off rather than on. Well, several of my students yesterday presented studies in which we compared by microarray the abundance of genes in the zona glomerulosa with the neighboring zona fasciculata. And this slide shows some which are between 10 and 25-fold upregulated in the ZG of human adrenal. And I'm going to just pick out two, the top one, a stem cell gene LGR5, and one which Drew Marzou talked to you about yesterday, DAC1. And in the individual plots from 20 adrenals, you can see in every case that the expression is switched on in the zona glomerulosa in green and switched off both in the zona fasciculata and in the aldosterone secreting tumour, consistent with the notion that these genes may be switching hormone off in the ZG, but disappear when aldosterone has to be switched on in the tumour. In both cases, the protein product is selectively expressed in the zona glomerulosa, as shown in immunohistochemistry, here at low power, here at high power, and in both cases, Lalo and Junwo have expressed the genes in adrenal cells and shown that they cause roughly 50% inhibition of aldosterone production. So we may suggest that one or both of these are responsible for the very sparse expression of aldosterone in the normal human adrenal. Well, I'm aware that was a bit of hardcore science, so I'd like to finish my talk by illustrating how the microarray findings, like the exome sequencing, also have had an immediate clinical impact. And this is apparent in the beautiful clinical story some of you heard Ada yesterday afternoon tell you about two women whose Conn syndrome presented in pregnancy. And I'm going to close with a short précis of her story. The patient who brought the explanation to light was seen in the last 6 to 12 months by my colleague in Cambridge, Fiona Corrett, when she arrived having had a termination in her first pregnancy because of severe hypokalemia. And Fiona made the diagnosis of Conn syndrome, and the patient went to surgery. And I met the patient first through her tumour, and the work of Alison Marker, our NHS pathologist, who now routinely does immunohistochemistry for a number of stains. And what she found was that the tumour had pure ZG cells, that it had very little of the enzyme that makes cortisol, was rich in the enzyme that makes aldosterone, and particularly rich in KCNJ5, which we have found to be a very good surrogate, better than B2 itself, for the presence of aldosterone synthase. And at this point, this rang a bell with me that this appearance and the presentation in pregnancy was very like a patient from 10 years previously who had also presented in pregnancy and had a very ZG-type tumour. So let me take you back to this slide that you saw earlier, where we found that 9 out of our 10 ZG-type tumours had mutations in either sodium potassium ATPase or a calcium channel, and you may have wondered about patient number 10, who was one of the few women in the series. Well, patient number 10 had a mutation in the gene ß-catenin, which is the hallmark gene of the Wnt signaling system. And so the obvious question now was, did our second patient presenting in pregnancy also have a mutation in ß-catenin? And the answer is yes. Here's the original patient down at the bottom. The newly presenting patient has another phosphorylation site mutation at the same exon of the same gene. So what's the relevance? And a second piece of luck was that this patient, PN, had not only taken part in our exome sequencing, but had also been part of the microarray comparing all the genes expressed in ZG and ZF. And so I was able to sort the results to see which gene expression was most different between PN and all our other adrenal patients. And the answer is LHCGR, which may not mean anything to you until I say it stands for luteinizing hormone choriogonadotrophin receptor, which is the receptor for the pregnancy hormone HCG. And on qPCR shown here, Ada found that both the patients and a third patient, whom we by this point discovered again with a ZG tumour and a mutation in ß-catenin, have a several hundred-fold increase in expression of the pregnancy receptor compared to all our other APAs, whether they were ZF-like or ZG-like. So I think the pregnancy presentation is explained, and to generalize now the potential for finding single gene syndromes within syndromes of possible clinical relevance. This particular mutation seems powerfully to predict complete cure from hypertension when the tumour is removed. So, Mr. President, complexity, Baron Platt might have remarked to Sir George, may be born of individual angels dancing on the head of a needle. And so let me summarize by saying that 60 years after the first patient with Conn syndrome, we find that hypertension is indeed commonly curable. His original patient may have been the tip of an iceberg. For many years, the more classical patient paradoxically had cells that resembled those in the normal adrenal which do not make aldosterone but make cortisol. And we now recognize the solution to this paradox, namely the common occurrence of small tumours which resemble the aldosterone secreting cells of the normal adrenal and have their own hallmark mutations. Well, my favourite slide is the last one, because I'm going to thank the people who've done the vast amount of the work, a young group of PhD students. And just as I think I learnt the ethics from my parents, who I mentioned at the start of my talk, that you work hard and then you go out and play hard, I think that's the ethos that the lab enjoy. This photograph was taken on afternoon after all of them presenting at the Athens International Society of Hypertension, and we went out and enjoyed ourselves at the Temple of Sunium. So thank you all very much for listening to me.
Well, Maurice, thank you for that. I personally think that's the best Pickering lecture I have ever heard. And I think it's amazing how he's managed to take such a complex subject and made it very accessible. And I think I now understand it all. This is definitely world-beating stuff. I think this is going to change our management of hypertension, and you're seeing something here which I think is going to result in a sea change in the way we manage patients. Now, traditionally, the Pickering lecture has no questions, but would you be prepared to take some questions? Are there any questions for Maurice after such a fabulous talk? Has he left any stones unturned? There's one question at the back here, and then... Okay.
Hi, I'm interested in some of the health economics surrounding this. How common is common? How expensive is a PET-CT scan? How feasible would it be to roll it out? So the implications to practice with rationing of care.
Yeah, well, obviously a very pertinent question, and this could resolve into short-term economics and longer-term economics. And depending on who I'm speaking to, I might start with the longer bit and say that patients with primary aldo seem to be a high-risk group. It can't be coincidence that some 25% of resistant hypertension is found to have primary aldosteronism, I think reflecting the damaging effects of aldosterone for many years. A point I didn't bring out is I've never seen a Conn's adenoma grow. They must have grown at some point, but probably in childhood or even earlier, and they've had years causing damage before we diagnose and remove them. So long term, I'd like to think that, especially if we can move from surgery to endoscopic ablation, it will be highly cost effective. In the short term, of course, there is a problem. The answer to your question, plasma renin, very cheap, probably no more than £15 a sample, but once you get into adrenal vein sampling or PET-CT, probably around £1,500, whichever modality you go down. And so the selection of patients is important, and as I think I hinted at a couple of points, there's really two extremes that we like to investigate. One is the younger patient, because there's such a high chance of curing them lifelong if we find a unilateral cause. The other is the older patient with resistant hypertension despite multiple drugs, or who won't tolerate the drugs. And in between, I think we have to say, if we're not certain we will cure the hypertension and the patient's happy on spironolactone plus minus semiloride, then the cost effectiveness is much more difficult to be certain about.
That was truly wonderful, but we've got about an 85% success at sampling, but interpretation is still difficult. Do you think we should be doing primary metomidate scanning now rather than even trying to sample?
Yes.
So is this the fast-track way of diagnosing?
Okay, but I should give a slightly longer answer. I mean, there are practical issues with the metomidate, which is carbon-11 has a half-life of 20 minutes. Yeah. So it can only be used where there's a cyclotron. Any of you in centres with a cyclotron, Kennedy, Cruikshank, put your hand up, please work hard on your nuclear medicine people to be interested. And we think there are at least half a dozen sites in the UK could set this up. And that's probably as many as are able to do adrenal vein sampling. So the UK should be able to do this. How we go from a situation where there's fewer than 300 patients having adrenalectomy a year at the moment to maybe a necessary 3,000 a year, it will be for discussion, but let's start where we are.
Any more questions? If not, Morris, thank you for an absolutely marvellous lecture. The Society is very pleased.